Welcome, everyone. Thank you for joining us for the DCMB seminar series and CCMB seminar series. It is my distinct pleasure to introduce Dr. Francesca Luca. Just to give you a little bit about her background, she did her undergraduate degree in biology at the University of Calabria in Italy. She also did a master's degree there where the topic was fine analysis of human molecular diversity in Italy, looking at SNPs on the Y chromosome. She did her PhD also at University of Calabria in molecular biopathology. Then she moved across the Atlantic and did a postdoctoral fellowship at the University of Chicago. Then in 2012, she started her own independent lab at Wayne State University, where her primary appointment is in the Center for Molecular Medicine and Genetics, and she has a secondary or a joint appointment in the Department of Obstetrics and Gynecology. Fast forward to 2018, she was promoted to associate professor with tenure in both of those primary and secondary appointments. Dr. Luca's research is at the intersection of statistical genetics and functional genomics, where she has reported a number of exciting manuscripts that I always follow her research, where she tries to understand how genetic variation within and across populations can influence complex traits in a gene environment, especially in an environment-specific context. She has editorial roles on numerous different journals. She's been a member on many different NIH study sections, and she's an overall exceptional scientist. So welcome, Dr. Luca. We are very excited to hear about your latest research, and I'll turn it over to you now.
Thank you very much, Steve, for inviting me and for the kind introduction. I'm gonna share my screen now. Sorry, there's the right thing here. Okay. There we are. Can everybody see my screen well? Okay, good.
Great.
All right, so I'm really excited of presenting my work here today. As like a next door neighbors of almost as we are. And so before going into the science, I want to give like a brief introduction of what my of my research program. So as Steve was mentioning, we are interested broadly in gene-environment interaction in molecular phenotypes and complex traits, and we've done work on cardiovascular disease and asthma, and I'll tell you more about it during my seminar today. We've also applied the framework of gene-environment interaction to study functional genomics of host microbiome interaction, and we also have an interest in characterizing the function and the evolution of non-coding variants. But today I will focus on our GBio gene-environment interaction work. And so, as we all know, especially in this audience, genome-wide association studies have been successful in identifying thousands of genetic variants that are associated with human phenotypic variation. And from integration of genome-wide association study results with expression quantitative trait locus mapping results, we also know that much of the phenotypic variation is actually due to changes in gene regulation, so that one of the most common framework for understanding the genetic variation role in complex trait is that we have regulatory variants that modify binding of transcription factors, which in turn affects gene expression and ultimately leads to inter-individual variation in complex traits. Now, however, we also know, and it's very, very, very well accepted, broadly accepted that genetic is just one side of the story, and that really what determines human phenotypic variation is not only our genotypes, but importantly the environment and the interaction between our genetic makeup and the environment we are exposed to. Despite this broadly and widely accepted notion, we have really very few well-characterized gene-environment interaction in complex traits. On the other hand, where we've been a lot more, this is blurry. Where we've been a lot more successful is in studying gene-environment interaction at the molecular level. And I apologize for the quality of this figure. It didn't look like that on my screen. But one of the approaches that's been most successful, both in our hands and in the hands of many other researchers, is using response QTL mapping as a way to identify gene-environment interaction in gene expression. And specifically, with this approach, what we do is considering two different context or treatment condition and performing QTL mapping, in other words, looking for association between genotype and gene expression and comparing whether this association is present in both condition or if it's condition specific. In this case, we should be able to see in theory that there's an association in the treatment condition between number of copies of the minor allele and gene expression, but we don't see one in the control. So this is what we would call a response EQTL or a context EQTL, or I think the most recent name is dynamic EQTL for this specific phenomenon. So leveraging these and similar approaches that I'll tell you about in a minute, we've been working on studying gene-environment interaction in molecular phenotypes and then using that information to learn about gene-environment interaction in complex traits. And so this is sort of the general framework that we've been working on, where we study molecular phenotypes that include chromatin accessibility, transcription factor binding, gene expression, and RNA processing. And then we look for genetic and environmental effects and then interaction on this gene regulatory mechanism, identify gene environment interaction in this molecular trait, and then use this information to learn about the role of G/E in complex traits. So an outline of what I'm going to be talking about today, I will be focusing on two main projects, G/E across cellular and environmental context in cardiomyocytes, and G/E's psychosocial experiences and asthma risk. So these are really two different aspects of our work on gene-environment interaction. And the reason why I've decided to present this is because this is work from two students that have very recently graduated from my lab, Anthony Finley and Justina Restak, and they completed this work during the last year and a half of pandemic. So I'm really, really proud of, their exceptional work. And, I was trying to pick between one of the two projects, and I was like, well, I'll tell you both. The other reason why I wanted to tell you about these two projects, because we actually just heard that we will be funded by NIH to continue both line of research. So we're actually looking for postdocs to continue the exceptional work that Justina and Anthony have been doing. And then hopefully I will have time to also tell you a little bit about some of the first follow-up work that we've been doing, incorporating single-cell technology into the study of G/E as well as transcriptional variability. And that's work led by Justina and Julong Wei, who's a postdoc in our lab. All right, so ideally to study gene-environment interaction, we would like to be able to measure all possible environments that we are exposed to in our life and precisely characterize our complex trait of interest and then look for these interaction effects. However, this is very hard for a number of reasons, including difficulties in measuring environmental covariates, as well as difficulty in defining precisely phenotypes. So one of the approaches that we've been using in the lab, as I mentioned before, is to focus on molecular phenotypes. And so in the two projects that I'm going to be talking about today, this first one is not only we're looking at molecular phenotypes, but we're also considering what we call cellular environment. So the idea here is that, as I said, ideally we would like to measure the complex environment that represent our lifelong exposures, but one difficulty is getting an accurate measurement of that environment. So we can go around this difficulty by focusing on cellular environment, which are essentially treatments that we can apply to cells. And so the advantage of using this approach is that we can tightly control the environment. Of course, the drawback, the limitation rather than the drawback, is that we can only look at one treatment at a time, meaning we cannot really model the complexity of the environment. And then once we've done this study, then we can go from the molecular phenotype to the complex trait in the organismal environment. So as I mentioned before, there's already been some work looking at gene-environment interaction focusing on molecular phenotype and in vitro treatments. And the most used approach is responsive QTL mapping. However, one limitation of responsive QTL mapping is that this approach doesn't scale well when we want to look at a large number of environments. And so in one of the first big projects that I did in my group, we really wanted to cast a broad net to get a better sense of the relevance of gene-environment interaction on molecular and complex traits. And so we, in that project, we focused on 250 environments. But if you want to do an QTL mapping study and say you want to use a minimum of 100 individuals, that would be 25,000 experiments that you have to do in the lab, which it's, you know, you don't want to be the student that has to do that experiments, right? So what we decided instead is to use a different approach, and an approach that is now being used by other groups as well, which is called allele-specific expression. So the idea here is that if we focus on a transcript where there's a heterozygous site, we can measure the expression of each alleles. And so in the absence of a regulatory variant for that gene, we will have a similar number of reads coming from each allele, while if there is allelic imbalance, then we will see an excess of reads from one allele compared to the other. Now, if we have a gene-environment interaction, that will be a pattern that will be specific to, for example, a treatment condition and will be absent, for example, in a control condition. And so this is what we've now got used to call conditional allele-specific expression or context-specific allele-specific expression. So in other words, it's a measure of gene-environment interaction. And the advantage of using allele-specific expression is that it can be measured even in a single individual. So we don't need to do the 25,000 experiments that I was talking about before, as long as that individual is heterozygous at variants in coding regions. And the second advantage is that because we are comparing the two alleles within the same individual, we are essentially fixing all the trans effects. And so we, those become negligible. And so we have power to detect allele-specific effects. So in our original study, we explored 250 environments, which were the combination of 50 treatments and match control in 5 cell types. And one of the major results was that 50% of the genes with gene-environment interaction are in genomic regions that are associated with complex trait that essentially contain GWAS hits. One limitation of this first study that we did was that we couldn't really disentangle what were the cell type specific effect compared to the treatment specific effect, which was we thought was a very interesting question. So in order to be able to better characterize allele specific expression and the relative contribution of cellular and treatment effects, we designed a new study, which is a study where we are using iPSC's technology. And the advantage of using iPSC is that now we have different cell types from the same individual. So in this case, we focused on 3 cell types from 6 individuals. So we had the iPSCs, we had the lymphoblastoid cell lines from which the iPSCs had been derived. And then from iPSCs, we differentiated cardiomyocytes. We then applied 28 treatments to this different cell types with a study design where we had 2 replicates for each treatment. So this is how we like more of a technical aspect, how we run these type of experiments in the lab. We have these like units where we have on each plate 3 individuals, and then in this case we had a replicate of the plate. And we were careful to process different cell types on the same day so that we didn't have a batch effect that might confound any cell type effect. All right, so of the 28 treatments that we used, we identify a strong transcriptional as well as splicing response for 12 treatments. And here I'm reporting the number of differentially expressed genes on the left and of differentially spliced genes. And so these treatments included a few different type of chemicals. We had chemicals that are broadly used, such as caffeine or nicotine, where there's like a broad exposure. We had some over-the-counter drugs like acetaminophen, some hormones, which they can be seen as a pharmacological treatment like dexamethasone, as well as an endogenous exposure, because dexamethasone is actually a glucocorticoid, which is the same molecule as cortisol, the same type of molecule as cortisol, which is one of the stress hormones in our body. Okay, so to identify gene-environment interaction through ASC, we first use an ANOVA model to detect shifts in allele-specific expression. And so we identify over 15,000 SNPs with ASC. We then use a linear model to specifically test for gene-environment interaction, where we have a cell type effect, a treatment effect, as well as an interaction between cell type and treatment. And so these are the numbers that we get. We find a very large number of gene-environment interaction, and these we have genotype by cell type, genotype by treatment, and genotype by treatment by cell type. So these would be treatment-specific, genetic regulation of gene expression that is also cell type specific. So as you can see, the largest contribution to gene-environment interaction measured by ASC seems to come from cell type. But one interesting aspect is also that we have quite a bit of signal, and we think that that's because of the specific study design, where we had essentially numerous replicates represented by the different treatments that we were using. So this is a bar plot of conditional ASC for treatment in red and treatment by cell type effect in green across the different treatments. All right, so one question in the EQTL mapping field is, how many EQTLs are out there? Do we need to still keep looking for EQTLs? And so the simple answer that we got essentially from GTEx is that pretty much each gene has an EQTL. So the question is, are we done then? Do we know all of them? Have we found all of them? And so the brief answer is no, I really don't think so. But now I'm going to try to walk you through why I think that's the case. So what we did here is ask the question of whether the gene-environment interaction that we find in our study, which in the end of the day are really evidence of the presence of regulatory variants, were already discovered in GTEx. And the short answer is no, that's not the case. But most importantly, what we find is that while genes that have allele-specific expression, which is not conditional on the treatment, are enriched in GTEx EQTL, both when we look at EQTLs in heart tissue as well as when we look at allele-specific expression, when we consider genes that have gene-environment interaction in our data set, we don't find an enrichment. And we have done it both for the cardiomyocytes comparing to the heart tissue in GTEx, but we also considered iPSCs compared to the I2QTL study, and for LCLs compared to the Juvarit studies, which is an EQTL mapping studies in LCLs. So it's not really just a cell type result, but it applies to all the 3 different cell types that we studied. And so almost 50% of the genes that we found with GBA were not found previously in GTEx. And so what this tells us is that Well, first of all, even with only 6 individuals, we are able to identify instances of gene-environment interactions and so regulatory or potential regulatory variants that were not captured in much larger studies. And the reason why we're able to do that is because we are looking at a variety of different environments. And so one of the main results of this study, we believe, is that it is important to continue exploring different environments because there's quite a bit of genetic variation out there that has an effect that is context dependent and needs to be found because it might be important for complex trait variation. So our study design also allowed us for the first time to investigate one feature of allele-specific expression, and so, in other words, how variable it is. And so more broadly asking the question of what are the factors that contribute to variation in allele-specific expression. And so the largest 2 components that contribute to variation in ASC are cell type and treatment, as you can see here. However, there's also a very large fraction of variation in allelic expression that seem to be due to unknown factors. This is what here I'm calling residual variation. Interestingly, this residual variance is actually correlated between cell types. So it's not just noise that we're measuring. It is something that is conserved across cell types and so might actually be biologically relevant. So to try and understand a little bit more about the biological features and the mechanisms that might contribute to variation in magnitude and variance of ASC, what we did was to look at different functional categories. So we partitioned essentially the genes by whether they were differentially expressed, by the features of their promoter, whether they contain a Tara box or they had a GC-rich promoter, whether the gene was differentially spliced, if it was tolerant or loss of function, mutations, the functional relevance of the gene, whether it was a gene responsible for cardiomyopathy in Mendelian diseases or whether it's a gene associated with complex trait in transcription-wide association studies. And we also looked at evolutionary significance of the gene by looking at the NDS. So then we asked whether the treatment component of ASC or the residual component of ASC or the mean ASC increased or decreased in genes that had these specific features. So here's an example for differentially expressed genes. We subdivided genes in non-being differentially expressed, being differentially expressed, or being highly differentially expressed. And as you can see here, the treatment component of the variance in ASC increases with highly differentially expressed gene. The residual component of the variance increases as well as the mean ASC. But in other cases, we find different patterns. So for example, for genes associated with complex trait, we find a decrease of residual variance and mean ASC when the genes are actually associated with complex traits. So what is the clinical relevance of this allele-specific expression and this gene-environment interaction that we've identified? So first of all, we looked at the cell type specific effects in the genetic regulation of gene expression in cardiomyocytes, and we found an enrichment for heart rate. It is interesting because we also looked at other traits of relevance for cardiovascular disease, and we actually only found enrichment for heart rate, which is the phenotype where there's a direct involvement of the cardiac muscle, which the cardiomyocytes are part of. We also identified 3 genes that cause Mendelian forms of heart disease and that have gene-environment interaction. And I'm showing here 2, one for nicotine and one for copper. So the reason why this is interesting is because one possibility is that these genes that are involved in Mendelian forms of heart disease may actually also be relevant for non-Mendelian forms, so common forms of heart disease, when the individual is in a specific environmental context. And then finally, we also partition irritability for several complex traits, and we found that there is significant contribution to the irritability of blood pressure for genes that have gene-environment interaction for vitamin A, copper, as well as zinc. So overall, this study showed that gene-environment interaction for a large number of cellular contexts can be detected by allele-specific expression. I showed you that the largest fraction of GBA is the result of cell type specific genetic effects, but there is substantial gene-environment interaction with treatments as well. And finally, I show you that treatment-specific genetic effects may modify cardiovascular disease risk, and this is shown by our study of irritability partitioning, as well as our overlap with Mendelian forms of cardiovascular disease. All right, so I'm going to be moving now to a project where, rather than considering in vitro exposure, we have actually been considering organismal level exposures. And so not all exposure can actually be modeled in vitro by specific treatments. And so here for this project in particular, we're interested in psychosocial experiences and asthma risk. And so we know that psychosocial experiences can affect health at multiple levels. So when I'm talking about psychosocial experiences, I'm talking about, for this specific project, 3 different types of experiences. One is what we call social relationships, such as, for example, social isolation. One is emotionality, such as anxiety. And then sort of kind of more objective measures of social experiences, such as socioeconomic status. So we know from other studies and from epidemiological studies and studies in psychology and sociology as well that these experiences affect heart disease, affect asthma. However, we don't know what is the mechanism. And so our hypothesis is that these psychosocial experiences affect gene expression and ultimately leads to variation in health in individual depending on their experiences. So the specific questions for this project is what are the contribution of genetic variation and the psychosocial environment to inter-individual variation in asthma symptoms and severity. And so the reason why we focused on asthma is because asthma is one of the leading causes of hospitalization as well as absence from school in children. And there's a higher prevalence of asthma in urban context and such as the Detroit area, the metropolitan Detroit area. So we did this project in collaboration with colleagues in the Department of Psychology at Wayne State, and they were really interested in understanding how the social experiences may affect asthma risk in children, while we were more interested in trying to understand whether there was a molecular connection that went through gene expression. So the name of the project is Aloft. It's a multi-year project that started in 2008, 2009. So I joined the project in 2014. And the goal of this project was to monitor and sample longitudinally more than 250 asthmatic children that had 10 to 17 years old and that lived in the metropolitan Detroit area. So one key feature of this study is that we have very extensive medical and psychological information. So our colleagues in psychology did an amazing job in recording not only questionnaires, but the children were also wearing an electronic device similar to an iPod. They would record sounds and noises in the household for like a minute at a time, and then they would decode these recordings to obtain an objective measure of family relationship and then social experiences that these children were having. On our end, we collected blood from this individual, from which we isolated DNA, we isolated RNA from leukocytes. We also had blood composition for a subset of the individual. We did test of glucocorticoid resistance, and we measured gene expression genome-wide as well as genotyping. Okay, so I told you at the beginning that one of the difficulties when dealing with organismal level type of exposure is that they're hard to measure. And so one of the first tasks for this project was, finding a way to deal with the fact that the data set was not complete. For some individual, we were missing the psychosocial information, in some cases because it had not been entered in the database yet, and in other cases because it was just missing, it was not recorded. But on the other hand, the other feature of this type of studies is that the measure of the psychosocial variable is inherently noisy, because if it's a diary, it relies on the child remembering the experiences of the last 2 weeks. Even the objective recording from the year is a little bit more objective, but there's still a certain level of noise that it's just inherent to the variable. And so we used GLMNet to define gene expression signature of this exposure. And so the idea here is that this would allow us to essentially increase our sample size by being able to predict the exposure for individuals for which we don't have a measurement. And on the other hand, because it's a transcriptional signature that is based on gene expression only, it is essentially a measure of the gene expression effect of that variable, which is denoised from other factors that might affect that variable. And so this is the cross-validated correlation for the different types of transcriptional signatures. As expected, we have, we had most success with a gene expression signature for blood composition, because of course it's the one that it's molecularly closer to gene expression. But overall, we were able to predict transcriptional signatures for several psychosocial variables as well as clinical variables. And so here's an example of the transcriptional signature for pulmonary function. So this is the transcriptional signature, and this is the measured for pulmonary function, and you can see there's a very good fit. To validate the transcriptional signature, we also had longitudinal data for these individuals, and so we derived the transcriptional signature at time point 0, and then we had the gene expression data at 1 year later and 2 years later for the same individual. And so here an example of variation in the transcriptional signature for the proportion of neutrophils, and this is the actual measured variable. And so you can see that in this case, there's also a very good correlation. So we then asked whether these transcriptional signatures are correlated with each other. So the idea here is that if with the transcriptional signature, we are capturing the effect of the exposure on gene expression, if we find that these signatures are correlated with each other, then it is likely that those two exposures or those two phenotypes actually share a molecular mechanism. And so what we find here is an example, what I'm going to show you here is an example where we have a transcriptional signature for socioeconomic status, which is correlated with a transcriptional signature for self-disclosure. Now, self-disclosure is to which extent the children were talking about their own feelings and their own thoughts to other people, which could be their peers or their family. So what we find is that these two psychosocial variables are actually also correlated with two biological variables, which is the percent neutrophils and the percent lymphocytes in the blood. And they are also correlated with asthma severity as well as pulmonary function. Now, we're not, we cannot say what's the cause and what's the effect here, but what we can say is that there seems to be a shared transcriptional profile that is associated with all these variables. And so the question, one potential interpretation that of course needs to be validated is that these psychosocial psychosocial experiences may have an effect on gene expression through changes in cell type composition, which ultimately leads to some of these clinical phenotypes that are important for asthma, which are severity as well as pulmonary function. So we then asked whether genes that are differentially expressed in response to these psychosocial experiences, whether they are known to be relevant for asthma as well as other allergic diseases. And so to ask this question, we used data from a TWAS, a transcription wide association study project that we had done with our collaborator, William Wen, here at University of Michigan. What you can see here in this plot on the left in this column, we have the psychosocial variable that connects to the gene that are differentially expressed as a function of this variable. And then on the last column, we have the complex trait. And so the connection between the gene and the complex trait is whether there is a significant TWAS result. Okay, and so the way to interpret is that now we have subjective socioeconomic status, for example, and the expression of this gene PFK-FB3 is associated with subjective socioeconomic status in our population. And now we also know that the expression of this gene is associated with hay fever, allergic rhinitis, and eczema. So we are now making connections between a psychosocial variables that is associated with changes in the expression of that gene, and knowing that the expression of that gene is actually important for this complex trait. And specifically for asthma. But we want to go one step further and ask, can we identify gene-environment interactions, or can we identify genetic variants where there is an interaction between genotype and the psychosocial signature, the transcriptional signature for psychosocial variables? So we use this model where we have the genotype dosage, transcriptional signature, so the psychosocial variable, and then an interaction between genotype and the psychosocial variable. And so this is the number of gene-environment interaction that we find. We find gene-environment interaction for cell type composition, which is not surprising. One way to think about our transcriptional signature is that it's similar to what people have been doing with the convolution of cell type composition in bulk RNA-seq data. It's essentially the same type of computational problem solved in a very similar way. And so this is not surprising. This is what you would call possibly a cell type specific EQTL. But what's interesting is that when you go here into this green area, these are the gene environment interaction with the psychosocial variable. And so we find 40 for socioeconomic status, 48 for self-disclosure. Another psychosocial variable for which we see quite a bit of important results is maternal responsiveness or parental responsiveness, so to which extent the parent responds to the child feelings, needs, requests, and so on. And then down here, we have the gene-environment interaction with more of the clinical variables. So the FEVs, the pulmonary function is a measure of pulmonary function. And so we also find gene-environment interaction for these more clinical variables. So I'm going to show you an example here of a GBA for this gene, GAS8, which is a gene that from TWAS we know is associated with asthma, and specifically higher expression of this gene is associated with, lower expression of this gene is associated with an increased asthma risk, as you can see here from this red triangle. So low expression is higher risk. And what we have here in this plot, so we have on the horizontal axis socioeconomic status for each individual. Each dot here is an individual, and then on the vertical axis is the normalized gene expression for this gene. And so what you can see here is that the A allele here increases expression of this gene only with individuals that have high socioeconomic status. And so because high expression of the gene is actually protective for asthma based on TWAS. Here we have essentially a potential interaction, not only on gene expression, but possibly on the disease outcome, because we know that high expression of the gene is protective for asthma risk, and we also know that the A genotype from our data, what you can see here, is associated with higher expression of the gene for individuals that have a perceived high socioeconomic status. So what is the what are the implications of some of these results, right? And so the implications are, kind of the broad implication is that when we consider disease risk, again, we should consider not only genetic risk, but also how that genetic risk can be modified by a specific environment. But then it also has more kind of ethical implication by, prompting scientists to say, okay, then we should consider individualized intervention where we should take into account not only the risk of that individual to develop asthma, but that individual may be at an extreme need of having psychosocial intervention to make sure that the family environment is less risky for a child that might be already predisposed to asthma. And so it's something that, like, as a geneticist, it's something, we don't think all the time, but honestly, this project really made me think a lot about what could be kind of the broad, social implication of the work we do, and how important it might be to then collaborate with people that are more, in this case, in the social science field, to think about targeted intervention that might improve health of children living in disadvantaged area. And so the summary for this second part is that social genomics approaches, which is an emerging field in science, in humans can identify potential molecular mechanisms that underlie differences in disease risk. So what I showed you is that psychosocial experiences and asthma symptoms are reflected in blood gene expression, and this in our study specifically in children with asthma. We observed that there's a sharing of transcriptional signatures between psychosocial and asthma traits, which might point to potential molecular mechanisms that connect psychosocial experiences to health. And finally, that genetic risk for asthma and other allergic diseases may actually be modulated by psychosocial factors. And I showed you an example of socioeconomic status, but we have several also with other psychosocial factors. All right, and I don't have a watch with me. I don't know how we're doing with time.
It's 4:45.
Okay, so maybe 10 more minutes. I wanted to tell you a little bit about what we're doing in terms of following up on these two projects. And so we are using single cell technology to study the effect of the treatment not only on gene expression but also on gene expression variability. So this was prompted partly by some very first papers that came out trying to identify variability QTLs, but also from our observation for the iPSCs and cardiomyocytes project, where we saw that there's this widespread variability both in gene expression as well as in the genetic control of gene expression. And so single cell technologies allows us to study in depth gene expression variability, because now we can measure the expression of individual cells, and so we can look at cell-to-cell variability in expression, both at baseline as well as in response to a treatment, so in a different environmental context. And so this is our study design. We are working on a subset of the individuals from the ALOFT study. So these are the children with asthma that I just told you about. And for this project, we focus specifically on African American children. It's in general a cohort that it's like over 70% African Americans. And then for this specifically, it's all 96 participants are African Americans. And we, in this case, we exposed the cells, the PBMCs, to treatments in vitro to simulate infection, bacterial infection by treating with LPS. We also used PHA, which is a T cell mitogen, and is supposed to stimulate mostly T cells as opposed to the LPS. And so we used 10x genomics, and we identify 4 major cell type clusters: the B cell, the monocyte, the NK cell, and the T cell. And we specifically decided to focus just on major clusters because we want to do EQTL mapping, and so we wanted to make sure we had enough power to detect EQTLs. And so in addition to treating the cells, with LPS and PHA, we also treated them with dexamethasone, which is, as I said before, a glucocorticoid, and so it's essentially a stress hormone. So we're not measuring psychosocial variables here yet, but we have an in vitro proxy for stress, which is dexamethasone. Okay, and so and as I was saying, the goal of this project is to look at the effect of the treatment, so measure shift in expression as well as in variability in response to the treatments. And so what we found is that these treatments, so both the immunostimulators, the LPS and the PHA, as well as dexamethasone, which, by the way, the role of dexamethasone, sorry, I should have said that before. The role of dexamethasone is of glucocorticoids in our body. So they are released by the hypothalamus pituitary axis in response to stress, but they are also released in response to an infection, not to activate the immune system, but actually to shut down the immune system to avoid going into sepsis. And so you probably all know that dexamethasone is also one of the few drugs that's being used to treat patients with severe COVID. And that's because one of the main, we now know that people that develop severe COVID essentially develop it's as a consequence of an exaggerated immune response. And so by treating those individuals with dexamethasone, essentially the immune response gets dampened, gets shut down, or at least reduced, so that the cytokine storm is prevented. And so what we found is that we have a strong response to the immunostimulated dexamethasone, not only in mean gene expression, but also in gene expression variability. And so what this bar plot up here is showing is the number of differentially expressed genes in response to the different treatments, the number of differentially variable genes, as well as the genes that have both differential mean and differential variability. And so what's interesting is that we find distinct effect on variability, which do not overlap to the effect on gene expression, or at least not completely. And so here are 3 example genes. This first gene here, TAP1 in T cell, we have the 4 different treatment conditions: LPS plus dexamethasone, PHA, and PHA plus dexamethasone. And you can see that there's an effect of the 4 treatment on gene expression, and it goes in the direction expected. So if the gene goes up with LPS, it goes down with dexamethasone, which is what we would expect. If it goes up with PHA, it goes down with dexamethasone, but there's no effect on variability. While here we have a gene where there's no effect of the treatments on the mean, but there is effect on variability. And again, even for variability, we do see the same trend where the immunostimulator and dexamethasone have contrasting effects, so have effects in opposite direction. And this is not just for this gene, it's actually a pattern that we see more broadly. And then this is a gene where there's an effect of the treatment both on mean and variability, but they go in opposite direction. So one thing, I didn't go into the details of how we measure gene expression variability, but essentially we control for the mean so that the effects of variability are actually independent from the mean. Okay, we then asked, given that we see these pretty strong response and variability to the environment, we also have a genetic control of gene expression variability. And so can we identify variability QTLs, and can we identify response variability QTLs? So the short answer is yes. So again, this is a bar plot where we have genes that are only E genes, so expression genes with an expression QTL, which is the darker part of the bar down here. Then we have at the top of the bar genes that have a variability QTL, so we call it a V gene. And then the part of the bar in the middle is genes that have both an expression QTL and a variability QTL. And so actually, the genes that have both variability and expression QTL are the smallest proportion of the three groups. So here are some examples, and I have down here what are the diseases or traits that these genes are relevant for. And I know these are like examples, but I'm going to tell you the message I'm trying to convey, and then we'll walk through the examples. So what it's apparent from these results, these example results, and more broadly from our results, is that there is clear effect on gene expression variability of the treatment and the individual genotype, and that this is for genes that we know are associated with complex traits based on transcription-wide association study. So the obvious question is then, is variability in gene expression another mechanism that might lead to variation in disease status? And so these are, as I said, three examples. This is a gene associated with asthma, and as you can see, there's an expression QTL, and there's pretty much no variability QTL. This is an example of a variability QTL, but no expression QTL. This is a gene associated with autoimmune diseases, so rheumatoid arthritis, multiple sclerosis, and psoriasis. And then this is a gene where there's both an expression QTL and a variability QTL, but they go in opposite direction. And so this gene, expression of this gene, is implicated in response to HIV infection. And finally, we also find response variability QTLs, which might take a minute to kind of wrap our head around the concept, but the idea is that these are genes where there's an effect of the genotype on the variability of the gene, but that effect is only present when, in this case, we add dexamethasone to the culture media. So cells that are only exposed to LPS, there's no effect of the genotype on the variability of the gene, but when we add dexamethasone, we have an effect of the genotype on the variability of the gene. And 11 of these RV genes, we call them, so response variability genes, were previously implicated in immune diseases in transcription-wide association study. And so this is really, I think, one of the very first times that we see these new genetic effects on gene expression, in this case, variability in response to a treatment. So, the question now, really exciting question is, what is their role more broadly in human complex trait variation? So the conclusion of this third part is that we detected effects of immune stimuli on gene expression not only mean but also variability, that there is genetic regulation of gene expression variability, and that it is independent on genetic regulation of gene expression mean. And so these results point to the question of whether gene expression variability may actually be an important additional mechanism for complex trait variation. So big conclusion slide here. So I showed you today 3 projects where some of the main findings are that gene-environment interaction measured in molecular phenotypes can actually uncover curative mechanisms for complex trait variation. That GBA effects may be latent in large EQTL studies in a single condition, but can be detected with smaller sample size if we analyze many contexts. And finally, that genetic and environmental effects on gene expression variability are a curative important mechanism for complex trait variation. And so I'd like to close by with a thank you slide to all the people that have contributed to these projects. So I have this figure of this picture of me. I was actually at a camp with my daughter a few years ago, Camp Howell. I don't know if you guys know about it. It's in kind of in between Detroit and Ann Arbor, but I look back at this picture and I was like, this is how I've actually been feeling since when the pandemic, started, and this is how I bet a lot of scientists have been feeling, doing acrobatic between dealing with everything going on around us, with our personal life, our families, and so on. And this has had like a gigantic impact on trainees and junior faculty as well. And so I really want to acknowledge the work of the students and the postdoc in my lab that, they've been able to do science despite everything that was going on around us. And so Anthony and Justina, who just both finished their PhD. Anthony is an MD PhD student who is continuing the MD part of his courses. Julong, who is a postdoc in the lab. Cynthia and Allison were a student and a postdoc in our group before, and they've moved on, but they contributed to the CM project. And Alan Mondiani, who is now a PhD student at the Weizmann Institute in Israel. He also worked on the cardiomyocyte project. And, last but not least, most importantly, as I know many of you know, my group is actually shared with Roger Picareggi, who is my colleague at Wayne State. And so this is all work done in very close collaboration with him. And I know, like Steve and many of you know, we share mentoring of trainees, and we have shared funding for some of these projects. And so certainly this is work. This is joint work with him 100%. And it's also been possible thanks to many collaborators, some of whom are there at U of M, William Wan and Xiang Zhu, as well as collaborators at Wayne State, Richard Slatcher and Sam Zilioli for the psychosocial project, and Yoav Gilad at the University of Chicago for the cardiomyocyte project, and our funding from NIH. And I think that's it. I'll be happy to take questions.
Hey, congratulations. That was awesome and exciting work. Thank you so much. If you were here in person, you'd hear a loud round of applause. I have so many questions, but I'm also meeting with you one-on-one tomorrow. So I'm happy to defer if anyone else wants to ask a question, or I'm also happy to start off with a question.
I have a question. You have multiple cellular phenotype, and they have sample size probably not that great. Do you have any power analysis for your interaction?
You mean for the allele specific expression?
Yeah,
that's a good question. So we haven't actually done power analysis like in the traditional sense, but I can tell you what we've learned as we were doing these projects. So our first project that I just glanced on, we had a lot more environments and we didn't have replicates because we wanted to, let me say it differently. We had a reduced number of biological replicates because we had 3 individuals. We didn't have any technical replicate because we wanted to maximize the environments that we were investigating. And we did find G by E. We did, we were able to say that they are important for complex traits, but they were in the order of hundreds. So 3 individuals, about 30. Contacts in the order of like hundreds. Now the new study, the one that I presented today, we had 6 individuals, so twice as many. But for each individual, we had 3 different cell types, and then we have 12 treatments.
Real pockets.
And we had technical replicates as well. And we found G by E in the order of thousands. Now, one could say, well, that might be because of the cell type you used, which sure it could be, but I'm more like inclined to think that it's because of the study design, which also allowed us different statistical framework. to identify this conditional ASC, this G by E, because now essentially what we could do was leverage the information across all these different replicates and all had the same genotype. So then you can really see departure from, you know, that 1-to-1 ratio in the ASC, and you had a much better estimation of what was the null, like what you would expect if there was no ASC. So I don't know if this answers your question directly, but you know, it's more what have we learned. More than, you know, what is like your theoretical expectation is, you know, what have we learned.
Thank you.
So I'll ask one for the second story about the psychosocial.
Can you hear me?
Oh, was there someone else?
No, I have a question. Skill.
Go ahead.
Thank you. Very comprehensive genetic environmental epidemiology study. Anyhow, I was struck by the GAS8 story and socioeconomic status. Where do you go next on that? I mean, it's a little hard to change people's socioeconomic status unless you're handing out cash. What is the plan for furthering that interesting observation?
Yeah, that's a good point. So, you know, short of not being able to, you know, do what you just said or change the politics or the social intervention, from a scientific point of view, we are actually doing a project now directly investigating how poverty may be associated with gene expression variations. So I'm doing a project with a postdoc who I'm mentoring, but she's actually from Morgan State University. And this is a project, actually we have a poster that we will be presenting at ASHG for people that are going there. This is the Handles cohort, which is a cohort that was assembled by the National Institute of Aging. And it's a cohort of individuals living in Baltimore. They were sampled, blood sample were taken 4 to 5 years apart. So there's 2 time points. And there's individuals living below the census-defined poverty lines and individuals living above the census-defined poverty line. And it's a pretty well sized cohort. It's if I remember correctly, it's like 250 individuals, and so 2 time points, which we are treating as if they were 2 replicates. And now we're looking for gene expression for genes where there's variation in expression between individuals living above or below the poverty line. And I can tell you we do find some. And what's interesting there is that there seems to be a sex-specific effect where genes differentially expressed in poverty in men are more related with immune system processes, while in women, they are more related with coagulation and wound healing and platelets. So it's very preliminary, but you know, and we don't have the genotype data yet, so we're not really looking at G by E yet. But if those results, you know, if these preliminary results are confirmed, I think it's a pretty, you know, I don't want to say definitive, but it's a pretty clear evidence that, you know, in that case, poverty status, which is a much more precise definition as opposed to perceived socioeconomic status, poverty status has an effect, which, I mean, of course, it has an effect on our health status, right? But the fact that there is, in addition to the fact that you cannot provide for your own care, that you are, you know, at a disadvantage for access to, you know, proper medical care and so on, there's also a direct effect on your biology, right? On your biological processes. I think that's going to be pretty interesting, and you know, it also opens the windows into like our biology and how, you know, how much influence there is from psychosocial factors and psychosocial experiences. So, you know, it's unfortunately it's not our place to solve those issues, but hopefully, we can point to effects. We can provide scientific evidence of the mechanism that social inequalities have on individuals' health, and hopefully that will make a more convincing case for the need to change certain political interventions and social interventions.
That's a very good statement. Very good. I look forward to talking to you tomorrow.
Me too.
Just as a quick follow-up to that, this like really profound implications of this work. And I wonder if you envision a day where someone has like an environment specific PRS where you can kind of predict which kind of areas of the environment they might be more susceptible to based just on their genetic variation, right?
Yeah, ideally that's really what we've been talking about. We've been talking about it with William Wen quite a bit, and that's what we would like to be able to do. That's we've written it in some grants as well. But that's kind of what I envision it. Hopefully, this field going to, and being able to have really individualized not only therapy, but also like preventive medicine, right? That is not just about, you should take this pill or this tablet because you are at higher risk for this. But, even just, we know exercising is good, right? But I think you can make a much stronger case if you tell people exercise is good, but, if you don't exercise, people that have your genetic profile and don't exercise have this much increased risk, and so as long as it's used in a good way, I think there's promise for not only exciting science but also translational potential.
Yeah, there's a lot of really broad implications. I think it's exciting. I have tons more questions, but we're going to meet tomorrow, so I'm going to withhold them now. And we're a little bit over time. But does anyone else have other questions or comments for Francesca?
Yeah, my question is on your emotionality and SAS. How was that determined? Is that based on the where they live, or is an individual based? And at the same time, my other question is, SAS is predictive of where you live. It's not just direct SAS, maybe other factors like, exposure to air pollution or other things. So how do you say the effect is really related to SAS instead of these multiple confounders?
Yeah, that's a good point. The confounders, for everything that we measured, we try to control for. But of course, there might be things we haven't measured and that therefore we didn't control for. So that's certainly one concern there. In terms of measuring socioeconomic status, this was subjective. For the project that I presented, it was subjective socioeconomic status, which is people were shown a ladder, and they said, on a scale from 1 to 10, where do you think you fit on the ladder? And so that's how it was. And it's I'm told from my psychology colleagues that it's a standard way of collecting subjective socioeconomic status data in the psychosocial field. The project I was mentioning before that that it's very preliminary and didn't show, in that case we did have census data, and so it was more objective, and it was also dichotomous. It was either below or above poverty, on the poverty line, while this one was continuous on a scale from 1 to 10.
Thank you very much.
Okay, we're a bit past the hour, and I know there's a number of folks that are going to meet with you tomorrow, so I bet you're going to get lots more questions. This is really exciting work. Thank you again for speaking at our seminar series, and I look forward to meeting with you tomorrow.
Thank you very much. Thanks for inviting me, and looking forward to the meetings tomorrow.
Bye.
Bye-bye.